Now we will move on to the second step of the FAIRe practice, which is to use the template and record relevant information. I will show some of the key information or formatting rules that you need to keep in mind when filling your metadata. For this demonstration, I will be using this demo dataset that you can see here. And this was sent out to you participants as a part of the workshop materials. And this is intentionally small and simple, but it covers the key formatting concepts you need to understand. And again, if possible, please follow along on your own computer. You can use either this dataset or even your own data. Let's start with the project metadata. This is the template that we generated together, the GBR 2022 project. And I'm going to start filling some of this information recorded by my name and ID. I use my ORCID ID, contact information, okay, now let's talk about this project ID related terms. This one, project ID, is already pre-filled by the FAIRe-ator. Project name, so project ID needs to be short and concise because it is used in file names such as this template, and then when you have an OTU table that is in, say, tab or CSV format, then you would also need to include this project ID in the file name. So all the separate files can be linked together. But this project name, you might put a full name of this project, for example, Great Barrier Reef 2022. And this parent project ID, this becomes very useful when you have a larger project where this GBR 22 is a part of. For example, I might have a parent project ID called GBR so GBR Metabarcoding Diversity project, and I would go sampling. I went sampling in 22, but I'm also planning to go next year under this parent project ID. And in this case, this data set is from 22, but next year I would produce another project metadata, sample metadata, and so on in the following year. I will use the same project parent project ID, but this project ID will be 23. In that way, these two separate data sets can be linked together. The users would know that these two data sets from 22 and 23 are from one large project called GBR Metabarcoding Biodiversity. Now let's jump into this PCR section, and I would like to explain a bit about these two terms, targetTaxonomicAssay and targetTaxonomicScope, because the difference between those two terms can be a bit confusing. The first one, this targetTaxonomicAssay, as you read the description, it is a taxa or species name targeted by the primers, probes, and other approaches applied in the PCR. So it's about this assay, what taxa this assay is targeting. So my fish is targeting fish, and then this one, crustacean. On the other hand, the scope is what you are actually interested in using this assay. So many cases, this will be the same as the target taxon assay. So, but just give you an example, I used this crustacean assay, but what I was really hoping to detect is malacostraca, such as crayfish, shrimps, and crabs. So in that case, you can now see that these two entries can be different taxa, slightly different taxa. But for my fish, what I wanted to detect was also a fish, same as the taxonomic assay. So hopefully this would clarify what some cases that these two can be slightly different. Other mandatory terms in project metadata are this primer forward and reverse. This should be quite straightforward. It's sequences of your forward and reverse primers in upper cases, and you would need to fill those for each primers. Now let's move on to sample metadata. And for this, we're going to be using this demonstration data, or you can use your own data and follow along. The first column in sample metadata is a sample name, and this is the sample name. It needs to be unique across the sample metadata. Here I'm going to copy paste from this demo dataset into the FAIRe format data. And you can see that there is a blank sample, the blank DI water control samples. We highly recommend to include controls in your metadata for the reproducibility and the transparency of your study. The next column here is a sample category, and it is a mandatory field and a controlled vocabulary. So you need to select from this vocabulary options, and first three of them are actual samples, the eDNA samples. So I will use sample here, and the last one here is a negative control, as I mentioned. Next columns here are negative control type and positive control type, and you can see these are indicated as a mandatory field, but there's a condition. If you look at this window, then it says the requirement level is mandatory if some category is negative control. Same for the positive control type. This is also mandatory if some category is a positive control. So in this case, my last sample here is a negative control, so I would need to fill this information, and it is a filtration negative control. When I was processing my water samples, I use a DI water as a blank, so this would be a process negative. The description of what this means, if you would like to look at, is indicated here. In this description, there is a Klimas et al. 2024, DOI is indicated there. So have a look at this table 2 in the paper, and it has a really good list of what type of negative controls people should be using and the purpose of it. So when you look at them, then you would understand exactly what these control vocabulary terms mean. Now moving on to the next mandatory field, which is the decimal longitude and latitude. This is indicated as a mandatory, and also it has to be, the format type is a numeric, it has to be decimal. As you can see here on this demo data, I originally didn't use a decimal degrees, so I would need to convert it. In order to keep the original record, there is also a term called verbatim longitude and verbatim latitude. So I would first copy paste these original records here, and then move on to the conversion to decimal degrees. In order to do the conversions, there are multiple tools available on the website and R packages and so on. But for this one, I'm just going to use this one. And I already entered my degrees, minutes, seconds of the latitudes and longitudes. make sure that this is negative because my coordinate is from the southern hemisphere. Then I will convert to decimals, and then I got this decimal latitude and longitude. So now I'm just going to copy paste this into the FAIRe metadata. These values I got here have 6 decimals, and the recommendation is to have a minimum of 5 decimals so that we can achieve the meter scale resolution as it's described in this description here. Having these verbatim values here is really important if your original record was not decimal latitude. If this coordinates doesn't make sense, sometimes it could be error, then you can always go back to these verbatim values and check if that was the error from the recording point or the conversion was somehow wrong. So this is why we have these verbatim terms here. Now, for the control samples, you often don't have a latitude and longitudes. And what do you need to do, especially if it's a mandatory field and you cannot enter the information? In the guideline, if you go to this guideline page, there is a section about missing values. So please go into that, and then there's a list of terms, missing value controlled vocabulary terms, to describe that data is missing and the reason why it's missing. These terms are from INSDC, Missing Value Controlled Vocabulary. So if you go to this link, you can get more information. But let's find the term, the appropriate term for this case, and it is not applicable because it is a control sample. So I'm going to copy paste this into this section. And you can always use these missing terms for the terms that are not mandatory, but that is not required. So you don't need to do it, but it is required to do that for the mandatory terms. The next mandatory term is a geolog name, and it has a quite strict fixed format, as you can see in this window. You need to start with a country or sea names that is from the INSDC country list. Then there will be a column, this region, comma, and locality. So in my case, I had the country name of Australia, column, and it was in the Queensland state. And the island name, I used as a locality, and that was Lizard Island. And again, for the control sample, it was not applicable because it is a control sample. The next column here is an event date, and again, it is a mandatory field. And as you can see in the description here, it needs to follow the ISO 8601 format, which has both year and time under one field. And as you can see, my original data set, it's not recorded in that way. So I'm going to use this verbatim event date and time fields to keep my original record, and then format them into ISO format for this event date column. Now, before I fill these verbatim columns, I also want to highlight about another date and time formatting challenge that we might come across and we need to watch out, especially with Excel. Sometimes even if you have it correctly formatted as date and time in Excel file or CSV file, it can change the format later when you read the file using other programs. And I found this happening in the output of the FAIRe Verifier tool that I'm going to demonstrate in the next video. There are a few ways to prevent this happening, but for now I will show you one way, which is to keep them as text format. And I will do this even for these verbatim dates and time columns. Currently, this date and time in my original demo data are recognized as a date and time. So if I simply copy paste this, try to make it to text. So I would do special paste options with the values. Then they will be converted to these kind of numbers, and we don't want them to happen. So I'm going to undo this, and then I will show you a different way. First, I'm going to create 5 new columns here so that I can use these columns to make date and time in text format step by step. And I'm going to call these columns as a date, text, time, text. And I'm also going to create this ISO format to prepare for the event date. So first thing I do is to create the date in the text format, and to do that I make a formula here, equal text, select this cell, and currently it is a date, month, and year. I will do the same for the time, text, hour, and minute. Now I'm going to convert this. At this moment, it is still recognized as a date. So I'm now change this to text, and you can see that this time it doesn't convert this into numbers. And the next thing I need to do is, because it is still a formula, I cannot just simply copy paste into another sheet. So I'm going to copy and paste as a value. Now this formula is gone and this is purely a text format. Then I can just copy paste into the verbatim event date. I will do the same for the time again. First I convert this to text, and to get rid of this formula I'm going to copy and paste special with values. So now it's not a formula anymore, and I can now copy paste this into verbatim event time. Now let's work to create this ISO format. So first I'm going to use this date here, copy paste into here, and format the cell as a date by using ISO format. Okay, now I want to change this one to text. So I'm going to do what I just did earlier. text, and this time it is year, month, and date. Now I'm going to change this format to a text and copy, paste as a value. The final step is to concatenate date and time and make it into ISO format. If you look into this event date description, you can find out the details of ISO format. So you need to put date, capital T for time, hours and minutes, and second as well if you have it. And then plus minus time difference from a UTC. In this case, I was in Queensland, which is 10 hours ahead. So I will put plus 10 hours. So let's do that here. Going to concatenate the date, capital T, time, plus 10 hours. Now you can see this as a correct ISO format. I'm going to format cell, change that as a text, and again it's a formula, so I'm going to copy and paste as a value. Now I can copy paste this into the event date. Let's check the format of these cells, and you can see it is text, which is what we wanted. Now let's have a look at the next three terms, which are mandatory fields. There are environment broad scale, environment local scale, and environment medium. These three terms are to describe about your sampling site and sample type. And these terms are originally from the GSC, the Genomic Standard Consortium Data Standard, such as mixes, and there is a strict formatting rules in the vocabulary that GSC implemented for these environment terms, and we are following the same rules. Those are all explained in the description. These three terms are mandatory in FAIRe checklist because INSTC such as ENA also have them as a minimum requirement. Let's start with this broad scale. So this is to describe the major environmental system of your sample collection site. And I mentioned about this field type that is a fixed format and also using a control vocabulary from ENVO term list. And so let's have a look at this ENVO site. The link is over there. If you want to copy paste, you will need to go into edit notes. Then I can copy and paste here, and it will take you to this ontology search website specifically for biome, and this is where we want to find the appropriate term for your broad scale. This ontology search website has thousands of words, terms that people created, and it can be a bit overwhelming at the start to navigate yourself, but once you get used to it, then it's a really good search website. So let's have a look at this biome again. And now under here, environmental system, ecosystem, there are 216 terms here. So when I click here in the biome, then it will give you more options. In my case, I would choose aquatic biome, and my samples were collected in a marine biome. I would stop here for my case, so I would use this marine biome for this broad scale description. So now I'm going to enter marine biome, but you will also need to see put the ENVO number, as you can see in the example, and the field type. So I will use a square brackets, ENVO colon, and then find the codes. That was this one here, and close with a square bracket. And control samples, again, I don't have that, so I put this vocabulary, control vocabulary here. And this is the broad scale. Now the next one is the local scale, and that is to describe about your location, the sample location, a bit more into details than a broad scale. And again, there's a link to the ENVO search website. And it will take you to this layer search engine. There are multiple layers, 500 plus, as you can see here. So I'm going to expand this and then find the terms that I'm after. So I will open up this layer, and from here, my case, it will be a fluid layer. And then if I look at this, it's not about atmospheric layer. I will go into liquid layer, aquatic layer, I suppose. I can see a few options again. I will go into marine layer. There are a few different zones, but my case was the sea surface layer. So I would go into this. So this will be the term that I want to put in my metadata. Again, I'm following this format where I would now enter these numbers. As you can see, there are multiple terms, and if you go into this layer, then you'll see lots of branches, and it can be hard to find, if it's your first time especially, find the appropriate term for you. So this search window here is actually really useful. So in my case, I would search something about surface water, so I put surface here. And then I can see a few list that has a term surface. You can also hit search, and then there are different type of surface terms here. Water surface could be useful, but at the bottom I can see sea surface layer. That's the one that I used earlier. So this is how you can also navigate this ENVO search engine. The third one here is the medium. and it is to describe the type of materials of your samples. So for example, soil, sediment, air, water, and so on. We put quite a few examples in the description, and hopefully this will give you the answer straight away for you. But if not, please go into this ENVO website again. That link is provided in the description. So in my case, I want to use the term seawater, which is listed in that description. So I'm just going to copy paste that into here. Oops, sorry. And again, the control sample is not applicable. Next term here is the habitat natural or artificial, and it's a Boolean term, so you need to answer them in 0 or 1. And this term is to describe whether your sample site is natural or artificial, such as aquariums and zoos. So for example, if your sample site was a zoo in Australia and detected giraffe DNA, then you would select 1, which means artificial habitat here. And this information helps biodiversity databases such as GBIF and OBIS identify records from artificial environment and exclude them from the species occurrence datasets. So GBIF OBIS users won't be seeing giraffe occurrence in Australia and get confused. But in my case, this was all natural environment, so I will put 0, and perhaps this control samples, it would be 1, that's artificial. Let's go into the sample size, and here we would use this example data set. So I would like you to pay attention that there is a sample size column here that is a numeric variable, and then a sample size unit that is a controlled vocabulary, and you can select from a range of unit terms. And in this case, I had the spore samples of this sample size. In the unit of liter. So here I will copy paste that and select liter. I would like to mention that there is a similar term called sample volume weight for DNA extraction, and that is mentioned in this description window here. And so this term is down towards the right of this sample metadata here, and that is the volume or mass of sample or subsamples that were processed for DNA extraction. And I will show you the difference between these two terms using this diagram. So for example, you collected 2 liters of seawater and filter that sample, and only use half of the filter paper for DNA extraction and kept the other half in a freezer. And in that case, then your sample volume will be 2 liters or 2000 milliliter. But then your sample volume for DNA extraction is 1 liter. So that is a case when these two values can be different. But if you use a whole filter membrane for DNA extraction, then the two entries under these two terms will be the same, 2 liter. So this sample size numeric variable, as well as many others, have a unit term here where you can specify what type of units it applies to this previous term. But other numeric variables, such as this one, sample storage temperature, This one, the unit is specified as a degrees Celsius. That's described in the description. So in that case, you will only enter a numeric variable, and then you won't enter anything about the unit because it's already been specified as a degree Celsius. Okay, so let's skip through these terms for now, and you will find the next mandatory term, which is the assay name. So here, the first row, it is already pre-entered by the R function, the FAIRe-ator, and in this case, I used 2 assays for all of them. If you want to list multiple entries, not necessarily only for this assay name, but in other terms, then you can use this vertical bar to list. In some case, maybe you only use 1 assay for some of the samples, but then 2 assays for the rest. Then in that case, you can modify like this. Another important point about assay name is that it appears in multiple places throughout the dataset, including the project metadata, experiment run metadata, and also file names for OTU tables. So it is very important to use the exact same assay name consistently across all tables and files. Be careful, for example, to avoid typos, spelling variations, or differences in upper or lower cases letters, and because these can prevent related data from being correctly linked together. After assay name, you will see a number of environmental variables here. And in many cases, you may not have values for most of these fields, and that is completely fine. While additional environmental information can make your data more useful for future data reusers, but you don't need to collect all of them just because they are listed in the FAIRe checklist. So you just need to provide the information that is available and relevant to your study. At the start of this environmental variable section, there are 3 terms here that are related to depth, and those can be a bit confusing to understand the difference between them, so I'm going to cover that now. These 3 columns are minimum depth, maximum depth, and total water column depth. The total water column depth refers to the depth of the seafloor or lake bed at the sampling location. In this example of the diagram, the total water column depth is 12 meters. The minimum and the maximum depths define the depth range from which the sample was collected. In this example, the minimum depth is 0 meter and the maximum is 2 meter. If you simply collected water from the surface as in another example, then both the minimum and maximum depths will be 0 meter. These minimum and maximum depths are not limited to water samples. They also apply to sediment and soil samples where the maximum and minimum depths represent the sampling depths relative to the local sediment or soil surface. These are all explained in the descriptions of these terms, but hopefully with these explanations, it is all clear to you now. This concludes the demonstration of completing the project and the sample metadata tables. There are additional tables to complete, such as experiment run metadata for metabarcoding studies and PCR standard and amplification data tables for targeted assay studies. I won't cover those in this video, but hopefully the examples that we have worked through together have given you a good understanding of how to navigate the templates. As you continue, remember to pay attention to controlled vocabularies and required formats, and to use terms consistently across tables. With those principles in mind, you should be able to complete the remaining tables with confidence. But that said, don't worry if you do make any mistakes, because the FAIRe-Metadata verifier or FAIRe is designed to help identify formatting issues and inconsistencies and guide you in correcting them. I'll demonstrate how to use the FAIRe in the next video.